welcome to The Explainer. Today, we're cracking one of the greatest detective stories in all of biology, the decades-long hunt for the actual molecule of life. So, back in the early 20th century, scientists had a pretty good idea where to look for hereditary information. They'd narrowed it down to the chromosomes. But here's the catch. Chromosomes are made of two different things, protein and DNA. So the entire field of genetics was basically stuck on this one huge question. Which one of these two molecules was the real deal, the one carrying all the instructions? It was a genuine molecular whodunit. To solve a case like this, the investigators, the scientists, first had to lay out some ground rules. They figured any molecule claiming to be the genetic material had to pass 4 specific tests. Okay, so here was their checklist. First, our prime suspect had to hold all the information, the entire blueprint for a living thing. Second, it had to be passed down from parent to child. Third, it needed a way to make perfect copies of itself. And finally, it had to explain all the amazing variety we see in the natural world. This was the list that was going to unmask our culprit. The first major break in the case came in 1928, and weirdly enough, it wasn't from a geneticist. It was from a microbiologist named Frederick Griffith who was just trying to figure out pneumonia. Griffith was working with 2 versions of the same bacteria. One he called the smooth, or S strain. It had this protective capsule that made it a stone-cold killer. The other was the rough, or R strain. No capsule, totally harmless. And at first, his experiments went exactly like you'd think. He injected the deadly S strain into a mouse, the mouse died. He injected the harmless R strain, the mouse was perfectly fine. No surprises there. But then Griffith did something kind of strange. He took that deadly S strain and killed it with heat, which made it totally harmless. Then he got ready to mix it with the living but also harmless R strain. So to recap, he took harmless living bacteria and mixed them with harmless dead bacteria. He injected this completely harmless mixture into a mouse, and the mouse died. What? This was a total shocker. It made no sense. When he looked at the dead mouse's blood, he found living deadly S-strain bacteria. The only possible explanation was that some transforming principle, as he called it, had passed from the dead cells to the living ones, like a ghost in the machine handing over new instructions. Now, this incredible clue pretty much just sat there for over a decade. Then, in the 1940s, a team of researchers, Oswald Avery, Colin MacLeod, and Macklin McCarty, decided they were going to figure out exactly what this transforming principle was, once and for all. Their method was just brilliant, pure forensic science. It was like a police lineup for molecules. They took the juice from the dead S bacteria and systematically knocked out one suspect at a time. First protein, then RNA, then DNA, and checked to see if the transformation still worked. And here it is, the moment of truth. When they used an enzyme to destroy all the protein, transformation still happened. When they destroyed all the RNA, it still happened. But, and this is the kicker, the second they added an enzyme that chewed up just the DNA, the transformation stopped. Cold. And you can see the proof right here. The dishes where they got rid of protein and RNA are teeming with transformed bacteria. But the dish where they destroyed the DNA, it's empty. The conclusion was crystal clear. No DNA, no transformation. The evidence was undeniable. That transforming principle, the thing carrying the hereditary instructions, it was DNA. The investigation finally had its prime suspect. But you know how science works. One piece of evidence, even a great one, is never quite enough. The final confirmation, the real smoking gun, came in 1952 from the super elegant experiment by Alfred Hershey and Martha Chase. They used a type of virus called a bacteriophage, which is basically a tiny little syringe that injects its genetic material into bacteria. Now, the virus has a protein coat on the outside and a DNA core on the inside. Here's the clever part. Protein contains sulfur, but DNA doesn't, and DNA contains phosphorus, but protein doesn't. So they use this to put a different radioactive tag on each part. They let these tagged viruses infect some bacteria. Then they threw the whole mixture into a kitchen blender, no, seriously, to knock the virus shells off the outside of the cells. And what did they find? All the radioactive phosphorus, the DNA tag, was inside the bacteria. All the radioactive sulfur, the protein tag, was left outside. This was it, the final, case-closing, irrefutable piece of evidence. The virus was injecting its DNA, and only its DNA, to take over the cell. The case was officially closed. DNA is the genetic material. So the culprit's identity was confirmed. But, of course, solving one mystery just opened up an even bigger one. How could this one, seemingly simple molecule possibly do everything on that checklist? How is it structured in a way that let it hold the blueprint for all of life? You can just feel the momentum building when you look at this timeline. For 25 years, every single discovery just laid the groundwork for the next one, all of it pointing towards one critical moment in 1953. Two last crucial pieces of the puzzle fell into place. A scientist named Erwin Chargaff figured out that in any sample of DNA, the amount of base A always equals T, and G always equals C. They were like perfect partners. At the very same time, Rosalind Franklin's absolutely groundbreaking X-ray photos showed that DNA was a tightly wound, repeating helix. And using those final clues, James Watson and Francis Crick put together their now famous model of the double helix. And suddenly, everything clicked. The two strands, held together by those specific AT and GC pairs, provided a brilliant way to store information and a dead simple mechanism for copying itself. It checked every single box. And just like that, the search was over. We had transitioned from just studying heredity to the age of molecular genetics. The book of life had been found. But solving that great mystery only revealed the next, even greater one. How in the world do we learn to read the language written in its code?